final session of the ALS Symposium is the Drug Development Roundtable. I'd like to thank Biogen, Cytokinetics, and Mitsubishi Tanabe Pharma America for being here today and being part of this conversation. Before we begin, I'd like to share a few guidelines we will be following today. During the registration process, we ask attendees to provide questions for this roundtable discussion. We will be answering as many questions as we can get to today. The panel is a discussion of thoughts, and no member of the panel is offering medical advice. Every roundtable member will have the opportunity to share their insight into each question asked if they would like to. We'll start with introductions. Stacy, would you like to go first?
Thank you, Michelle. I'm Stacy Rudnicki. I'm Vice President of Clinical Research at Cytokinetics, and I had been a director of an MDA ALS clinic for over 20 years at the University of Arkansas. So I know your organization well, and thank you for including me in the call today.
Thank you. We're happy to have you. Toss, would you like to introduce yourself? You are muted. Still nothing. In the meantime, why don't we have Gustavo introduce himself?
Okay, I hope you can hear me.
Yes, we can.
Okay, cool. So my name is Gustavo Soy Sambrano. That's my official name. I am the VP for Med Affairs at MTPA. I am a neurologist. I've been with MTPA for about 3 years, more or less. And perhaps also with Stacy, I'm not sure you mentioned, I think you mentioned MS clinic as well. I have a lot of background. My main background is in MS, multiple sclerosis, both clinical and research. But I've been in ALS for now quite some amount of years, and of course my target is to try just to help the patients and the caregivers and those who suffer from this disease, and to see how we can help them get access to better or more options for treatment. So thank you for having me on tonight.
Thank you. We're happy to have you. Toss, do you want to try one more time?
Hopefully this works.
Yes.
Sorry, the meeting chose the wrong microphone for me. My name's Cochran. I am a neuromuscular neurologist and a medical ethicist. I was at Brigham and Women's Hospital for 17 years before coming to Biogen, where I am now the global medical affairs lead for ALS.
Thank you for being with us today. I will quickly just go into the first question. What are the biggest challenges in ALS for drug development? Stacy?
I'm happy to start. I think one of the biggest challenges has to do with the heterogeneity of the patient population. Now, it is a disease that can start in an arm, in a leg, with changes in voice, with changes in swallowing. It changes quickly in some patients and very slowly in other patients. And when you mix all of those people into the same clinical trial, it can become challenging to identify that the drug is effective. So I think the fact that the patient population is so unique individually that it does make it challenging to run clinical trials. Mitsubishi Tanabe, I'll give a shout out to you guys because you're very smart. You looked at your trial that had failed and identified responders and did a subsequent trial that was successful by changing your inclusion criteria. And I think, trying to decide the best patient population is where, I think all of us are starting to look at that. And I'm hoping that leads to success by identifying who is the right patient population for a given trial.
I'm not
See any additional
myself,
challenges?
yeah, it's up to you, but I guess I'll go first for Toss because he's unmuted somehow. Still, I see the microphone on mute. So now you're good. I will agree with you, Stacy, deeply. I think it's such a heterogeneous population, and it's not like one thing fits everybody, right? And that's what we're trying to do. I thank you for the kudos to our development on regards to at least trying to define what's the population that may benefit the most from certain therapies, right? This will give a higher chance to success at the end. And I think what's also important is after, let's say, a drug is or a therapeutic agent is successful in phase 3 trials, it's more about finding out more data beyond that population that was part of the clinical trial. That's where databases, registries, open label extension studies, but I'm not sure, the open label maybe not, but yeah, registries and those kind of data sources may really help trying to really understand what's the real benefit of the drug beyond the study patient population. That's the one comment.
I so agree with you there, because it's what's the best population to be able to identify a benefit in a reasonable amount of time in a reasonable number of patients. I don't think that means they're the only patients who might benefit from the drug. It's just our goal is to find drugs that work and get them into patients. And the quickest way to find drugs that work and get them into patients may be that you do have to select your trial population. It may work in people outside that trial population. And it just would take many more patients or much longer, because perhaps some of those patients who are progressing slowly just need to be followed longer to see benefit. So I totally agree that subsequent studies using country-based databases or clinic-based databases will be really useful to see what happens when you take the drug into that real-world evidence that we'd like to better appreciate and understand.
So Stacy and Gustavo, I agree completely with both of you, and I'd add that one of the things that the field could really use is a more objective early marker of axonal injury and neurodegeneration, right? Than the tools that we currently use for stratifying our studies, or more objective than the covariates that we use to control for, and having a biomarker that could tell you precisely how fast is this patient losing neurons would help you conduct more efficient trials. It would give you more confidence in the outcomes of those trials, and it would allow you to more quickly look at candidate compounds and figure out, is this a promising compound? And so I think the field does need some further evolution in that direction, and it would go a long way towards helping with that heterogeneity problem, Stacy, that you started with, and hopefully that's the direction that we're moving in.
We've looked for biomarkers. There's a lot of interest and enthusiasm for biomarkers. Unfortunately, I don't think we have the perfect one yet, or maybe we have a biomarker that's good and we don't quite know how to apply it. But we've had trials that have been positive with failed biomarkers, but good clinical benefits and vice versa. And we need one that, matches up. In my mind, the biomarker needs to move in the same direction as the clinical response. So, it certainly is an area of huge interest, and I'm sure we'll get there. I just don't know when we're going to get there.
Yeah, one comment I just want to make, and I don't know what you think, Stacy or Taz, is also the clinical endpoints and the outcomes we use in clinical trials, right? Sometimes, we all use the ALS FRSR because it's what is accepted from their perspective and what's been validated. but sometimes I feel this tool may fall short in trying to really determine what's the clinical impact or the benefit of a therapeutic agent, for example. So I think that's a point where perhaps we also can help, trying to help validate different endpoints, trying to perhaps create some endpoints that are more sensitive together with the biomarkers as well. If we decide to move into the clinical phase and see how they behave, because yeah, sometimes I think just the ALS-FRSR may not be as sensitive as we wish, and sometimes we may miss things that are still important in the clinical trial, and then the drug will seem like a failed drug, and then we close the research.
Yeah, I think the subjectivity of the endpoints that we use, even beyond the FRS, there's some subjectivity in vital capacity measurements, right? There's some influence of expectation and effort and fatigue that influences those outcomes. And so we either need to move toward a multi-endpoint model for assessing whether a drug works or not, or we need objective biomarkers that can help support those clinical findings.
Certainly there is a lot of interest in digital biomarkers right now that maybe, I think they hold a lot of promise, but they're not accepted by the FDA. They're helpful as exploratory endpoints in trials. And there's a lot of work that goes into validating those. And if the companies that are developing some of these tools, are interested in pursuing a program that the FDA offers that would basically qualify them as an outcome measure, that would be fabulous. That's something that hopefully will happen with at least some of these tools, like the voice apps, the things that measure fine motor on a digital surface, the accelerometers. I know that there's one accelerometer that's been gone through the qualification process in muscular dystrophy, for example, but it takes years to do it. So it is not for the faint of heart to get some of those digital biomarkers actually accepted as an appropriate outcome measure by the FDA.
Thank you all. Another question that came from our audience is, what can you tell me about CNTF from the early 90s? And I believe, Stacy, you were going to take this.
Yeah, so I'm the only person old enough on this call to remember the days of the trophic factors. So, and actually my very first ALS clinical trial that I was an investigator for was IGF-1, which is kind of in the same category. So there was CNTF, ciliary neurotrophic factor. There was BNDF, BDNF, brain-derived neurotrophic factor, and IGF-1, which was insulin growth factor. They were all used in the 90s in clinical trials. And the trophic factor kind of gives away what was thought in terms of how they would be effective. They were felt to be, they're substances that our bodies make naturally, but they were supplemented, given as an injection, usually a subcutaneous injection, which is just under the skin. And they were studied in clinical trials thinking they would help protect the health of the motor neurons that were struggling. And unfortunately, none of them panned out. CNTF is noteworthy because we just were talking about ALS FRSR. It was not the primary endpoint in the trial, but it was one of the secondary endpoints. And it was actually developed by a group working in the CNTF clinical trial. So at that point, it was the ALS FRS, there was no R, and it was 10 items instead of 12 items. But that was, kind of a hallmark change in the ALS world. So historically, it has that added perspective that it started the trend of moving towards using the functional rating scale is the outcome measure that many subsequent trials used.
Thank you, Stacy. Where do you see ALS treatments in the next 5 years? Gustavo, would you like to start? Oh, I believe you're on mute. Nope, still on mute.
Can you hear me?
There you go, yes.
Okay. So I think there is a multiple array of clinical trials now going on at the moment. I think some are preclinical trials, and some of the medications are very close to regulatory submission or being approved, or I don't want to talk specifically about our product, but recently approved, right? So I think there is a lot of movement going on in that area. I think there is also the topic about how can we be more efficient on clinical trials and get drugs to patients earlier. I think the Haley trial is one example, and I think they are making a great effort to ensure that at least some of the therapies that need further research are being studied in a very efficacious way, and they are able to decide early enough if a therapy may be efficacious or not. Another point, and I know those you are perhaps have been involved a lot on this, is of course gene therapy, right? As we make more advances in ALS, there are more discoveries we make, and then we understand more the pathophysiology and what may be causing this disease, and all the different heterogeneity that comes with the disease as well. So I think there is a lot of effort on that area. So what I will say is, in general way, I'm extremely optimistic. I think when I was trained as a neurologist, I had riluzole, and I kept having riluzole for many years. And besides trying to be an advisor, somebody to talk to for the patients and their families, and try to see what else I could do for them that was very limited, there was not much I could offer them. And I think now the horizon is much more positive because of what I've said and because of what the discoveries that have been made to understand the disease further. So that's perhaps my comment there. And perhaps those you want to add on your research or your experience?
Yeah, well, thank you, Gustavo. I think you touched on genetically targeted investigational drugs, and one of the changes that I think that may prompt, and I think we're seeing it already, is an increasing rate of genetic testing in people with ALS. And I think there are many people who feel like it is going to need to be part of the standard of care for ALS. And that is increasingly true as more and more trials have specific genetic targets. And eventually, if we have genetically targeted therapies, things that work for genetic subsets, then it really will have to be part of the standard of care in order not to miss out on something that could help. Speaking of something that could help, I think it's unlikely that in the near future we're going to have something that is the treatment for ALS. ALS is so heterogeneous that a multi-drug approach is likely to be the future of ALS care once we have more than a handful of things to use. And so I think it'll be difficult to sort out what's the right, once we have many things to choose from, it'll be hard to sort out like who needs to be on which drugs and in which order and in what combinations. But that'd be a good problem to have if we have some more therapies that are available.
I will say that, I already alluded to the fact that, I took care of ALS patients starting in the late 1980s. So I've been in it for a long time. And I used to naively tell patients, well, if no one else in your family has had ALS, you don't have a genetic form, because that's what we thought. I was not an outlier by saying that. I was the norm. And then it became, as we hugely expanded our understanding of how complicated the genetics of ALS are, that we now know that, that statement isn't true anymore. And now many neurologists routinely test everybody, all the patients who are willing to get genetic testing do go ahead and get genetic testing. Now we have over 30 different genes identified that are felt to either be the cause of or increase one's risk of developing ALS. So clearly that is hugely expanded targets for us. To me, it makes perfect sense, at least in those patients with those gene mutations, to target therapy when as it becomes available to their genetic mutation. So I think that certainly brings a huge amount of hope. And certainly in the SMA world, we've made amazing gains in treating SMA, and hopefully we'll get there with the genetic forms of ALS as well. The other thing that I think is so different is we have an explosion of companies interested in ALS. That it's an embarrassment of riches in terms of how many companies now are looking at ALS and looking at it from lots of different angles. So it's not like everybody is pursuing the same modality, etc. People are coming from it in very different ways, and that will lead us to have, I agree with you, multiple drugs available, so that you're not just addressing one of the things that we know go awry in ALS. You could potentially be addressing multitudes of them. So it's certainly, having been on enough of these calls, meeting new people. I've met Toss before, and I've seen Gustavo on other calls, but there are lots of new people now who are involved in ALS from very, very early to later stage trials. And that explosion and enthusiasm by the biotechnology, big pharma, and big pharma communities, is a huge shift and a welcome shift.
Yeah, the only comment I want to add to it is also we're learning more and more about the importance of having the patients and the advocacy groups being involved also in helping us trying to design trials that perhaps are looking to what is really important to those patients and those families, right? And I think the more and more we get involved that way, I think the probability of success within the next 5 years, maybe less than that, maybe more than that, who knows, is gonna be much higher, right? We have the science. We just need to make sure that the most important part of the ALS community, which are the patients, have a venue to perhaps express what is important to them, and we can try to include those in the clinical trials. I think that will go a long, long way in order to be successful.
It's hard to say that something good could come from COVID, but one of the good that came from COVID is we all looked really hard at our trial designs and made them more patient friendly. And you talk about patient input, and patients have long said you need to make your trials more patient friendly. And now we have, because we were, we had to COVID proof them. What happens if somebody gets COVID? What happens if you can't do a vital capacity in an ALS clinic? We found workarounds now. We were forced to, unfortunately, because of a pandemic. But I don't think those benefits that we reaped, getting lab at home, doing telemedicine visits as part of a clinical trial, they're here to stay. They're not going to go, I'm convinced, they're not going to go away because the pandemic hopefully will go away. But that is a huge, I think it's a huge benefit to also drawing in more patients who might not be able to get every month to a clinic visit, but they could get every 3 months to a clinic visit and have the intervals handled via a telemedicine call. That's, I think, is a huge plus that will make a real difference in being able to enroll patients more broadly than perhaps broadly geographically than we have in the past, because that becomes a less daunting undertaking for them to sign up for a trial.
Stacey, you touched on SMA a little bit, and based on what we've seen with different levels of progression of ALS, some live with it for a year or even 10 years. Do you think that we are going to start seeing different types of ALS like we've seen different types of SMA in children and adults? And Toss, I'll let you start off answering the question.
Sure, this is probably appropriate for me since I'm familiar with Spinraza for spinal muscular atrophy. I think my answer is yes and no. So in the sense that we are getting smarter over time about the pathophysiology of ALS and particularly, you know, genetic subtypes of ALS, we are going to have to start thinking about subtypes of ALS as slightly different diseases, right? And for the genes that we do understand the best, it's certainly fair to say that there are definite distinctions between different genetic subtypes. And eventually, you know, hopefully there'll be therapies that are targeted at those genetic subtypes. Having said that, the genetics of SMA are vastly simpler than the genetics of ALS and the pathophysiology of ALS. So SMA is about 95% of it's caused by one gene going wrong, and it can only go wrong in a few different ways. You can have no copies, one copy, two copies, three copies of the relevant gene, the relevant backup gene. And there's a really strong correlation between how many copies of that gene do you have and what's happening to you. You know, when do you develop disease? How severe is it? And that's so much simpler than ALS is, where even if you know you have a genetic mutation, that doesn't always mean you can predict when's your disease going to start? How severe is your disease going to be? How quickly will it progress? Will your sibling with the same mutation develop it at the same time? It's so much more complicated than that. So it's a much more difficult problem, but I think we are getting smarter over time about those subsets.
Does anyone have anything to add?
So we already do have some of those subsets that we've identified by phenotype, by how the patient clinically manifests. So some people have traditional ALS. Some people have primary lateral sclerosis, or PLS, a much slower disease than ALS. Some people have progressive bulbar palsy, where they have predominantly signs and symptoms related to chewing, swallowing, speaking, breathing, because the bulb, which is the old term for an area of the brain called the brainstem, has been predominantly the area in which motor neurons have been lost. And then there's progressive muscular atrophy. So we do have some of those segregated because of what you see clinically. And you have some of that segregated because there are genes that are playing a role. So we already have some of those differences, but what we don't have yet is how we might address those differences in different ways. And maybe there are some drugs that are going to work really well if you have progressive bulbar palsy, and others that may work really well if you are a traditional ALS patient. Those are things that we don't know yet, but certainly things that are definitely worth exploring, and maybe things that we learn over the course of time, particularly as we get more and more patients in clinical trials.
I don't know what else to add, to be honest. I think goes back to the heterogeneity of the disease. And one thing that may be simpler in SMA, for example, is really much more complicated to apply, implement, and get positive results in a population like ALS. I think Toss expressed that very clearly. And also we have to manage those expectations, right? We learn more each day. And sometimes we learn so much that sometimes we don't know what to do with that much that we have learned, right? That's kind of the premise. So we continue to explore and try to find ways to try to separate those different phenotypes. I don't know if that's the best word to use right now, but and see how we can approach each one of those, at least by groups, and then see if there's anything really we can offer to those specific groups. But it's just so heterogeneous that the difficulties are much larger.
On the topic of getting access to drugs in development to individuals with ALS, how can we do that, knowing that it's not just, it could be a geographic issue, a language issue? How do we tackle all of those barriers?
This is another problem that's complicated and doesn't have just one answer in terms of how to get more people involved in research. I do think that there is a definite importance in geographic diversity in our studies, and making sure that it's not, whenever possible, we have representation from multiple countries, multiple ethnicities and racial backgrounds in our studies, so that they're properly representing the diversity of ALS itself. On the other hand, that introduces heterogeneity into the study, which increases the challenge of detecting a difference. And so there's a little bit of a tension there, but certainly we should prioritize breadth of conducting our studies as broadly as we can. And I guess the other thing about trials is that they have to be our priority in terms of increasing access. We should be prioritizing increasing access to trials and extensions of those trials, and so that whenever we're talking about access to an investigational drug, we're also talking about how to generate data from that access, right? If we're not generating data on drugs that haven't been proven yet, we're not really living up to our primary duty, which is to try and find treatments that actually work.
Yeah, I think yes, find a way to get more patients from different areas to get into clinical trials. It's extremely important. I'm a huge fan, and I don't know if that's going to answer your question, but I think we all are, of the extension studies. I think you briefly touched on that, Toss, and how important they are. One is because sometimes you finish a phase 3 study, and finishing the study doesn't mean that the drug's going to be approved the day after the study is concluded, right? That's number one. So what are you going to do with those patients that willingly participated in this study, and then all of a sudden you don't want them to be out of drug until at least there is a regulatory approval, and then you can commercialize the product, right? So I think it's important for that reason, but also important in the open label extension trials, because sometimes it provides more safety information and more reassurance about the product's safety in general, right? And you may see things that maybe you didn't have the chance to see while you were doing the initial phase 3 study. So I think that's extremely important. The other piece is, I mean, sometimes we talk about expanded access programs and those kind of things. That will depend on the product that's being looked at. Sometimes when it's a new product, I think, and there's no other option out there, that should be a priority, and see how you can get access to those patients that really need it while the product is being studied. On the other hand, sometimes you have a product on the market and a similar product being looked at, perhaps that program will not be as critical, but an open label extension study will be more critical just to understand more the safety and perhaps the efficacy of a product. So that's perhaps the comments I wanted to make on this topic.
I agree. An open label extension is both for the company and for the patient of benefit. For the company, you get that added information about safety, what's referred to as durability of effect. You know, does it continue to seem to offer some benefits over a prolonged period of time? But also the patient finishes what is almost always a double-blind placebo-controlled phase. So some patients are going to be on placebo or something that's in an inactive tablet that's not going to provide benefit. Yet they did all the things, came to all the visits. And so it's acknowledging the commitment the patient made to provide that information to the trial was hugely important, and permitting them to then enter a period of time when everybody knows they're getting a drug, I think it's a commitment to the patient and a way to, basically be fair that they have spent a lot of energy and effort getting the answer for others, and to be able to have them continue on the drug while everybody else goes through the trial, because the first patient in is also the first patient out. And so they can potentially, be waiting for a year or even 2 years for everybody to get through the trial. And so they continue to receive drug until the answer is known. And hopefully the answer is positive, and they'll continue to receive drug until it's approved and available at the local pharmacy.
Is MTPA, Biogen, and Cytokinetics doing anything specifically to get patients and families more involved in the ALS drug development process?
So I guess, who did you mention first? MTPA? I think that's why I just jumped first. But
yes, go ahead.
So I volunteer, I guess, first. So the answer is yes. And of course, we are doing now, focusing more after we got the products approved, which is a little different now, into post-marketing or phase 4 clinical trials. That's the example I want to pose here. We spoke about biomarkers, the importance of biomarkers, and how they relate to the disease progression, and perhaps how we can use them in the future to help with clinical decisions, right? And with a treatment plan. So there's a study that we are running, actually, that we created basically with the support of the patient ambassadors. I think Rich Betlak is very involved with this group as well. And basically the study was designed with critical input from those representatives and those patients. Within the selected biomarkers, the duration of the study, and other things were basically done because of that feedback. Now, we wish to get them more involved. So I think if we start a new development program, for example, I think, like I said earlier in this panel, we need to really get the patients involved sooner than later. They really know what they need. They really know what's important to them, and I think the clinical trial should be able to deliver some sort of outcomes that are really relevant for them. And I think the FDA is also looking for that as well. The FDA is looking, is that clinically meaningful? It's this endpoint, something that is going to provide some real benefit to the patients. And they look into also those kind of inputs, if those inputs were given to the companies. So absolutely. And the more we do, the better, I think.
So in our current enrolling trial, which is called Engage ALS, we had two groups before the protocol was finalized, we had input from 2 groups of people living with ALS and caregivers of those living with ALS. So one was from Niels, part of that Rick Bedlack, trying to educate people living with the disease and caregivers more about advocating for ALS, and then a group from ALS. And they provided great insight into how to make the trial more patient-friendly. We also have a person living with ALS on our steering committee for our current trial. And then the advocacy arm of Cytokinetics has an advocacy council, advisory council that meets every 2 to 3 months, where we discuss various things and get a lot of feedback. So that is certainly an area that I think we've all become more cognizant of how important it is to engage the community, not be, narrow focused, what outcome measures, what's the statistics, what's the p-value, but really thinking about those who have to come to those clinic visits and the caregivers who help make that possible.
And I guess I'd say, at Biogen, we do similar things. So Stacy and Gustavo have mentioned sort of involving the advocacy community in early discussions about study design, the value of certain endpoints, the clinical meaningfulness of endpoints and things. And, we also think about other things like measuring outcomes with caregivers, right? So caregiver quality of life assessments, exit interviews for participants in our studies to sort of ask them about their experience of being in the study, feeding back reports to participants after a study's concluded and the results are known. We try and summarize the learnings from that trial and give it directly back to participants in the trial as a way of honoring their the participation in the study and their willingness to give their time. I think we can always do more. We could always do better. But I think particularly in ALS, the voice of the patients and the caregivers is really critical for us to get better at measuring things that matter and measuring things that really do tell us what's going on in the disease, rather than sort of what we imagine. Makes it challenging to do without hearing the voice of the people with ALS.
One comment, I think, Stacy, you mentioned the COVID era, right, and the pandemic, what we have learned, the good and the bad and the terrible things we learned. But if there is something good, for example, I think we're also trying to work with the patient groups on trying to identify devices, things we can do that will increase the level of comfort for any patient who is willing to participate in a clinical trial. Trying to validate in-home spirometry, for example, and so we can use it from home instead of having patients coming to the clinic every so often, which by the way could be difficult, right? We're talking about people that may live several hundred miles away from a clinic, and it's a whole day ordeal to go and visit a clinical trial site to get the assessments and the outcomes. We're also trying to see how we can, the biomarkers again, how we can draw the samples from comfort of their homes instead of having them come and get the samples taken in a clinic. So we protect the patients, and who better knows that than the patients, right? So that's something that we are trying to incorporate as well. And again, like I said, this is kind of something we didn't look at perhaps 2 or 3 years ago that deeply. Now we're really extremely involved. And I believe in your company, Stacy, and Tosh, you are also extremely involved on how to make life easier for these patients and make sure that we succeed in the clinical trials, but they also are comfortable and they're still supporting the science. So that's the comment I wanted to make.
Thank you. A question that came in from our audience was, if all drugs finally get approved, do you believe they will be prescribed altogether? Is there a limit?
I'm going to let you answer that one because that's another easy one, but I'll answer too.
Well, I'll say the short answer to that is yes, there probably is a limit, but it mostly comes down to either practical considerations or questions of safety, right? So there's lots of practical reasons why there's a limit to how many drugs you can be taking at any given time, and it may end up coming down to sort of what you're willing to tolerate. I know I can't take a medication 3 times a day. I can't manage it. So taking multiple medications on different schedules would be really, really challenging, and you'd have to make a decision about whether it's worth the trade-off. The mechanism of action of the drugs is a consideration. Are these 2 drugs competing with each other, or is it possible that there be a synergy between them? So it'd be even more important to take them together. And then finally, safety, right? Is one drug going to interfere with the other or create some safety risk? And so, like I said, in the future, this is going to get more and more and more complicated. How do I know which drugs to take and in which order, in which combination? It's going to be a good problem to have, but it could get really thorny eventually.
I was the person who responded to Michelle and said, yes, I think that's an okay question, because I think it's something that we all wonder about, It is, which would be so exciting if we actually would have the problem. If we had more than 2 drugs to be talking about, that would just be a huge plus for everybody on this call. I think we'd all be excited. I doubt that 5 drugs are going to be approved in a given year. Like, I'm sure this is going, I'm convinced more drugs will be approved. They will come out gradually, which will probably then kind of force part of that question, the answer to it in part. The 3rd drug or the 4th drug, which mechanistically is not working like riluzole or edaravone, it will be easier. And then I think once you get past 4 drugs, you're probably going to start it's going to fill the person up. There's just so much you can tolerate medicine wise. So some of it will be practical, almost certainly some of it, like a gene therapy, obviously that's for the person with the gene mutation. So some of those choices will be natural because of what the drug is designed to do. A lot of it ends up coming down to safety, and then it ends up coming down to what insurers are willing to pay. And that's a big unknown that none of us fully understand. So, but I think at least in the short term, they'll be allowed in combination, and as more and more drugs get added on, there's probably going to be some filtering that takes place.
Yeah, I think oh, go ahead.
Oh, I'm sorry, Gustavo. I was just going to add that we should also keep in mind that as more things get approved, it's going to make conducting studies on the next thing more and more complicated. And detecting an effect in somebody who's already on 3 or 4 drugs is going to be really challenging. It's going to make study design harder than it already is.
Yeah, I do agree, and I think when I think about multiple drugs, I kind of go back to some of my prior years in multiple sclerosis and how we had no drugs, and then we had all these 10, 12, more than 12 medications, different mechanisms of action, different safety concerns, and then how were we supposed to choose even which one goes first, and then which one goes second, and which one goes third? Can you mix them up? Mm-hmm. And I think this also perhaps is something that will be important if you're to discuss treatment guidelines, right? Keep in mind that, physicians, we try to do our best, but sometimes we need also people that guide us through the process, right? It's one drug plus another, a fair thing to do for a patient. Is one drug plus another even better? Sometimes, like some of you already have pointed out, if you add too many, maybe damaging also, may get more safe, patients may get more safety issues and things that we don't want to see, for example. So I think a piece that is important, and I would say the professional societies perhaps should begin to think about guidelines in collaboration with the advocacy organizations as well. We cannot be involved in those kind of documents and activities, but some others can. So I think that could help on trying to make these decisions as the future, I think, gets more complicated, but I think it gets brighter too, right? More options is better, I think. So that's important to consider and see how we can really mix them up, if we need to mix them up or not.
Thank you. I probably should have asked this first before we started, but during Dr. Lomenhorth's presentation from UCSF, she presented on research and clinical trials earlier. She began the presentation by talking about how she got interested in ALS in the first place, and it was because she had neighbors growing up who had ALS. So it piqued her interest. She went to med school and specialized in neurology. So can each of you share about why you chose to focus and work on ALS?
I'm happy to start. So I went to med school thinking I was going to be a general practitioner. Nothing could have been further from the truth because I became an ultra specialized neuromuscular physician. That neuromuscular change in my life occurred because I fell in love with neuromuscular patients as a resident. It's what tweaked my interest. Thomas might know David Chad, because he was at the MGH for a while. He was my original mentor. He was, and it is still, a wonderful human being and a really amazing neuromuscular physician. So many of us have people like that. And then I went to the University of Arkansas, where I was the first neuromuscular trained physician in the state. And yeah, underserved for sure at the time I went there. And a patient with ALS approached me and said, would you establish an ALS clinic? And it was like, sure. Little did I know how much work that would entail, but it was an opportunity to change, to change a state's way of managing ALS. And it was a huge joy in my life. Hard to leave that, but I partly left because I wanted to expand that reach at the impact I might be able to have. And hopefully, as new drugs get developed, one of them will be ours that is successful. So that's how I landed in ALS.
I'm happy to go next. And we're all neurologists, so I think we're going to end up having similar stories about why this is so important to us. One of the jokes that medical students will make about neurology is that, well, not even a joke, but they'll say, I don't want to go into neurology because there's nothing you can do for some of these diseases. It's so depressing. And for me, rather than being depressing, I kind of thought of it as all the more reason to go into it for two reasons. One is that even when somebody's got a disease like ALS, which is just, it's so cruel, it's so devastating. It takes somebody who's healthy, often in the prime of their life, and just robs them of their motor function. And it just couldn't be much worse. And there's not a lot that we can do in terms of modifying the disease course. But on the other hand, taking care of somebody who's in that situation and doing it well and delivering the news to them in the right way and supporting them through that journey and palliating their symptoms, it's hard to think of something more rewarding than that. Helping somebody through one of the worst things that can happen to you is actually very rewarding in and of itself. Then the other thing to say about it is, hopefully, when I was starting, I was thinking, okay, I've got a 30-40 year career ahead of me, I hope. And the very fact that there's nothing really good for this disease is all the more reason to be there when it does arrive. hopefully by the end of my career, we'll be seeing, more and more really effective therapies. And who wouldn't want to be in on that? And so then my transition from academia and neuromuscular practice into my current role was about, okay, I think there's a possibility that we're getting close to these therapies, and I really would like to help make sure that happens and be there when it does. So it's sort of a natural evolution for me.
So I'm not sure if you want to hear my history, but I started medical school because I wanted to be a dermatologist. Okay, so whatever. I finished med school, and then I wanted to be a plastic surgeon. And then I said, you know what, I'm not for that. I don't like standing up in ORs, and I just get desperate. I like to move. So I decided to go into internal medicine. And when I was in internal medicine, I decided to switch to neurology. So that's how I ended up in neurology. I really found my passion when I was doing my rotation in neurology, when I was doing my internal medicine residency. I finished my neurology residency, and that I'm going to say something, but I probably the younger neuromuscular neurologist in this call, in a way that my focus really in ALS has been in the past 3 to 4 years. My focus initially was on my fellowship in immunology and multiple sclerosis, and I did that for many years. But I got to a point in MS where a lot of things were already discovered. There were many options, and ALS always was kind of lagging behind, right? It's still it's such an unmet need, such a deterrent condition. So I was thinking, where can I make the biggest impact now that we have so many things in multiple sclerosis? And that's what I really decided to come into and support the ALS community. One thing I will say though, my first patient as a medical student, my very first experience was a woman with ALS. And I remember their families, and I remember her family, I remember the people around her. And I remember my professor, we didn't have many answers to give to the family or the patient. We just say, okay, just we'll see what we can do. Be patient. This is what's going to happen to you in the next few years. And that's a patient that I have remembered all my life. I have her face right in my brain. And every time I talk about ALS, I remember her and how limited we were at that time. And how much can we do right now for those living with ALS in this year and age, and how much more we can even do farther in the future. So I think that's why I decided to come to ALS as well.
Well, thank you so much, all three of you, for your honesty and candor in that response. We are wrapping up our drug development roundtable. Do you have any final thoughts for our audience today? Stacy?
Michelle, just to thank you. It's been fun to be on this panel. I've enjoyed hearing Gustavo and Tas's comments as well, and hopefully the audience learned some things that they didn't know before they started.
Likewise, thank you, Michelle. Thank you to the MDA and the audience. I probably speak for Stacy and Gustavo when I say this is one of the more fun things that we get to do, is to talk to the community, and I'm happy to do it again in other forums.
Yes, thank you very much for the invitation, and it's been a pleasure to meet you both, Stacy and Michelle. I hope I get to see you sometime in person somewhere. I expect better things to happen now, but I want to thank the patients too for their commitments and support to science. We could not do this without them. And again, has been really fun, and to be honest, we have been in calls probably all day talking about many things, and there's no better way to end up our day with this conversation. So thank you very much.
Yes, thank you to Toss from Biogen, Stacy from Cytokinetics, and Gustavo from MTPA. We appreciate you so much for all that you do for the ALS community, and we will let you get back to work to finding a cure for ALS.
Go back to our families.
Oh, it is the end of the day, so back to your families, but tomorrow start again. Back to work.
Yeah, thank you.
Thank you. I would also like to thank our guest presenters from yesterday and today, Mark Fisher, Ted Campbell, Shivangi Thorne, Dr. Catherine Lomenhorth, and Sarah Feldman. We appreciate your time, expertise, and everything you do for the neuromuscular community. We would also like to thank our ALS Symposium supporters, Biogen, Cytokinetics, Mitsubishi Tanabe Pharma America, Apellis, Ionis, and Hillrom, now a part of Baxter. We would love to hear your comments about today's symposia topics. If you have a smartphone, open your camera and point it at the QR code on the screen. A webpage will pop up with a short survey on today's symposium. If you do not have a smartphone, once the symposium is over, a screen will pop up with the survey as well. Everyone who fills out the survey to completion will be included in a raffle for a $50 gift card. If you attended yesterday's session and completed the survey, you will be entered into the raffle twice. For additional questions, please email them to mdaengage@mdausa.org, and we will follow back up with you. If you are new to MDA through this program and are diagnosed with a neuromuscular disease under MDA's umbrella, or are a loved one of someone who is diagnosed, we encourage you to stay engaged with MDA. You can do this by visiting mda.org/join and completing a short form. This will help you keep up to date on the latest information and education to help keep families informed in the neuromuscular community. This concludes the 2022 MDA Engage ALS Symposium. Thank you very much for attending and have a great rest of your evening.